missing here, guys? Okay. First, I'd like to thank my fellow organizers for allowing me to speak to you today about some of my data. They hardly put up any argument at all. It was very kind of them. The other thing I'd like to do before I get into some data is take a second to comment about microarrays. Most of the proteomic discussions we've heard today, and I believe it holds for the rest of the conference, involve mass spectrometry, which is a very powerful technique, especially in as much as it shows in vivo consequence of manipulation. If you, for example, make a mutation in a dub, the mass spec experiment will tell you the biological consequence. Where it falls short a little bit, I think, though, is in telling you direct effects. You don't know, for example, that the mutation you made didn't affect another dub or another E3 that then changed your levels of ubiquitination. The array excels in showing you direct consequence. I can absolutely guarantee that your dub or your E3 is putatively a substrate, putatively act on these substrates, and then you can take the leads and find out their biological relevance. So that's where we excel on the arrays. Like a mass spectrometer, the microarray can be a very versatile platform. There's a lot of different questions we can ask with a microarray. And the two that we will have time to talk about today are at the top and in black. One is, given an E3, can we identify substrates for that? And that is a very well-established use of the microarray. This is something we've published on. Paul, fellow organizer Paul Andrews, published on this recently too with SMRF1. A number of other groups have published it. It's very well established. What I'm going to spend most of my time today talking about, though, is the second one, given a dub, identifying substrates. Now, this, of course, presupposes a pre-existing post-translational modification. I can't remove a ubiquitination event until I've deposited it, so we will talk about both, but briefly, because at least with the E3 substrates, because that is more established. Just by way of quick mention, I do also want to point out that we can go in the opposite direction. For this crowd, we are probably more interested in the dubs and the E3s and finding out what the rest of the proteome's involvement with those enzymes is. But the majority of people that do basic science research actually approach the ubiquitome from the other direction. They have their own protein of interest, and they're more commonly going to ask, what's the dub, what's the E3, speaking to my protein of interest. And this is something we can also do on the microarray. Given the protein of interest, we can identify the dub. That was actually some data presented last year by Karina Balut from Dan Devers' lab at the University of Pittsburgh, and it's coming out any minute now in FASIB Journal. We are together actually with Karina and Dan right now working on the other side of that coin, taking their protein of interest and trying to identify the E3s. And then there's some other things we can do. We can screen enzyme activity in a multiplex format on the microarray. We published that last year. And the list of things that can be done with antibody arrays is extensive. I won't even begin to go down that path. There's a number of variations we can play. Whatever application you want, we can use single lysine mutants in the absence of linkage-specific reagents. It's not ideal, but it works. We can change the E2s, a number of different things to customize the experiment. Okay, so first, E3 substrate identification. This is a schematic illustrating how the process works. And basically what you do is whoa, can I go back? Thank you. Basically you start with a naked array. The proteins have no pre-existing modification, and they're stuck to the glass, immobilized on the surface. In solution phase, you add your ubiquitination machinery, ubiquitin, E1, E2, E3, and ATP, and you let the magic happen for an hour or so. You then wash away everything in solution phase and probe with labeled antibody against ubiquitin to visualize the covalently attached ubiquitin, thus identifying putative substrates. And the gain in signal here is what you're looking for. Those are my putative substrates. Okay, the first step when you want to do this type of experiment is make sure that you're using a good E2, one that will at least work with your E3. And this data comes from Mike Edens at Progenra, provided to me. He had done this screen for me. It's actually a kit marketed by my company, LifeSensors. It's known as the E2 profiling kit. It's a very facile assay. It's an ELISA-based assay. I believe we're up to about 25 different E2s now that you can quickly screen for activity with and without your E3. And basically the way it works is the wells of the plate are coated with a capture reagent specific to ubiquitin chains. So auto-ubiquitination or free chains will be captured If the E2 works with your E3, we pull it down and you quickly detect it, and you get the profile for which E2s are working. The next thing you do is, of course, take your E2 with your E3 of interest and move on to the array. In panel A, I don't know if you guys can see it better than I can, there should be a signal right next to that arrow, and that is a pretty decent signal. It's not the most robust on this particular array, but it showed up nicely. Circled for you is some controls. You have some biological normalization at the data analysis step we'll talk about in a second. Panel B, this was actually suggested from the data analysis. This particular substrate, it does not show up very robustly. This I'm showing because this is about the cutoff of where we drew our substrates. Anything less intense than this did not come through and get identified as a substrate. And panel C is a particular section of the array of one of the subpanels that had absolutely no signals and no substrates present in it. So that's more or less the range you see on the microarray. And the first thing you should notice is specificity. You don't just ubiquitinate any lysine. You do see a very rare event here and there. And that's important if you want to get real targets. Step 2, this takes a little bit longer. This same with a mass spec type experiment. You generate so much data, it takes a while to analyze it. So you spend most of your time at your computer doing smart things to make sure that the next follow-up steps are maximized for their likelihood of following up. You don't want to follow and pursue a target that's not likely to be real. So on the left, we're not going to go through this in any great detail. It's in the paper if you're interested. But I'm showing the process of low S normalization for technical noise. And in D, we're looking across the entire data set, and you can see some noise due to either the print tip or the location on the microarray. After the low S normalization in panel E, it goes away, and your data looks very clean. You can then come in and ask for biological difference. I was happy to see today already some talk about gene ontology clustering. That's the next step. However, this list, which I know you can't read, you don't need to, is traditionally where a lot of proteomics experiments end. And I can tell you firsthand that it gets very frustrating to look at this type of list and take a guess. You know, what's my real targets here? Where's the false discovery? How do I follow up on this? And people, unfortunately, used to assume that the protein listed at the top, number 1, must be your best, and the one at the bottom must be your worst. Turns out your false discovery rate, wherever you drew your cutoff for identifying substrates, your false discovery rate tends to be very constant across all of these proteins. In other words, the last one on this list is almost just as likely to be real as the first one. So gene ontology clustering is the next thing we do. It takes less than a day, and it's a very powerful way of asking, is there anything in my data set overrepresented from what chance alone would predict? This particular study comes from the E3 Project 1, which was known to be highly expressed in brain, so we were very encouraged to see brain development be one of the clusters that showed up. The only known or suggested substrate for that E3 was involved in apoptosis, so having a category come up as anti-apoptosis was also very encouraging. But that's basically the process for E3 substrate identification. Now, moving on to dub substrate identification, the fact that identifying E3 substrates works poses a problem, right? If I need to reverse a post-translational modification, just seeing one here and there is going to be a problem. So we need to overcome the specificity problem, which is illustrated here. You see a panel of substrates listed on the left, a number of different E3s, and they all tend to have their own specificity. We need to overcome that. The way we've done it so far for this proof of principle study has been to take a mix of E3s, throw them all together, and drive a more complete, you don't need to go back, drive a more complete ubiquitination of that array. And this is the data. This is not unlike the E3 substrate identification, which was done on a protein array containing thousands. That particular array had 2,000 human proteins. The current iteration of that product has 10,000 human proteins that we get to look at. This is not that array. This is a small microarray we'll talk about later that we produced at LifeSensors. It's one family of proteins. And what you can see in the complete reaction indicated with the plus sign is that almost every single protein on that microarray got ubiquitinated when we used a mix of E3s. And withholding any of the components of the ubiquitination reaction ablated the signals. Okay, so the final step, of course, then, once you've quote-unquote fully ubiquitinated your array, is to add a dub and see where you lose signal. And so in this case, we're looking for a loss of signal as an indication that we've seen dub activity. This is some of the data. Again, the small family protein array. There's 16 identical arrays shown here. In the corners, in the upper left and lower right, we mock treat with a dub, and as you would expect, you retain your ubiquitination, almost full ubiquitination of the arrays. Into each of the other subarrays, we've added individual dubs, and sure enough, lost some signals in some cases. USP 2 core and USP 8 core being nearly complete loss. Not a lot of specificity in those core domains. One reason you might not lose a signal, you would predict, is that you don't have enzyme activity. If the dub's not active, of course, it's not going to cleave anything from your array. So this, without going through it in great detail, shows that, in fact, most of these dubs are active. Circled for you is the lack of activity observed in this assay for AMSH core. This assay, by the way, is the CHOP assay licensed by LifeSensors from Progenera, pioneered by Dr. Craig Leach. Very easy, facile assay for dub activity. But it did not show us any activity for AMSH. We were curious. We thought we make enzymes pretty well over at LifeSensors, so we took it into a different assay. In this case, the CHOP data is shown again. On the left, on the right, we are looking at diubiquitin internally quenched fluorophore substrates. On the top, we're looking at the one that the diubiquitins are linked through K63. On the bottom, they're linked through K48. And as expected, predicted by the literature. AMSH core shows specificity for the K63 IQF. So the take-home lesson from this is that your dub activity probably depends greatly on which assay you're using. So use the right assay. Okay, so this is taking the data we just saw and organizing it for you visually based on the substrates, which are listed on the left here. And the mystery will be lifted in 1 second. The substrates, the reason I didn't want to get into it earlier, is that the substrates are dubs. This is a microarray of proteins containing deubiquitinase enzymes. So it gets a little bit circular and confusing, but keep in mind that the substrates, again, are always listed here for you on the left. After ubiquitinating these dubs, we then come in solution phase with the dubs listed across the top. Either no dub, where you preserve your signal, 2 core, 8 core, which takes them all away, or these others. And again, we're looking for a loss of signal. So the first thing that stood out to me, and the black box here is ubiquitin. It's control on the array. We spotted monoubiquitin on the array. You would expect to pick this up in any experiment with or without dub. So yeah, the red boxes are showing you one of the first things that stood out to us, namely that these active dubs are not capable of deubiquitinating themselves on the array. It does not look like you've got that self-trimming amongst the dubs. This is the same data, but now with all of the other substrates. These are the ones that just popped up were ones we have not tested yet in the solution phase to see if they remove signals. And we saw some interesting things looking at this data. Again, we're looking for a loss of signal. Forget for now the core domains that show, almost complete removal. What you want to do is look across left to right at the signals and see where you specifically seem to lose some, and that's what I've highlighted here. USP 15 is potentially a substrate for 20, and USP 20 is potentially a substrate of USP 15, and USP 7 is potentially a substrate of USP 15 based on the loss of signal. Ataxin 3-like is the first of that really dark section. Doesn't seem to be touched by anyone. We haven't found a dub for that one yet. And there's some other interesting things. 21 core, interestingly enough, even though it seemed to take away everything up here, was not able. It's definitely different than 2 core and 8 core in that it cannot remove the signals from those particular dubs. The previous data was all based on detection of the array with FK2, millipore antibody capable of seeing monoubiquitination and anything higher. This is the exact same experiment in which we, instead of detecting the array with FK2, chose instead to use our tubes, tandem ubiquitin binding entities. These are specific for polyubiquitin chains. And so now what we're able to see is if any of the chains were able to be trimmed that we formed on this. And Ataxin 3-like finally did show us a substrate that looks like PL-Pro, the SARS coronavirus dub, appears capable of trimming a chain on Ataxin 3-like, whereas nobody else seems to touch it other than, again, the core domains of 2 and 8. Interestingly, some of these dubs are capable of trimming free chains. In other words, ubiquitinate the ubiquitin that I spotted, and the signals go away for 28, 21, PL-Pro, PLP2. et cetera. Also, interestingly, that ability corresponded perfectly with the ability to remove change from USP4. But it's not an ability that goes beyond. In other words, you can remove the free chain, you can cut that, and you can remove a chain from USP4, but the presence of signals on all these other dubs says that there's something about them that prevents their change from being trimmed down. Okay, so that's enough pictures staring into the clouds, as I call it. We're now going to move into the section where I try to show you that some of the stuff we found on the array is real, and it's not just artifacts of the array. So the first question was, are the dubs in fact ubiquitolated? And so we ran the reactions in test tubes, E1, E2, E3, ubiquitin, ATP, and throw in a dub, run it out into a gel, and probe with tubes, tandem ubiquitin binding entities. They should only see polyubiquitin chains. And no dub control shown on the left. You do see some auto-ubiquitination in this experiment, but it's greatly enhanced ubiquitination in the presence of any of these other dubs. This was through a loop to that first gel, though. This data is something I've been thinking about a lot lately. What we've done here is raised antibodies against two of the dubs, JOS-D1 and UCHL5. And in both cases, we are running the complete reaction, E1, E2, E3, et cetera. We're running the reaction again in the absence of an E3, and I'm running just the protein alone, the protein being JOS-D1 or UCHL5. And what you can see is that in the presence of the complete reaction, we detect a specific monoubiquitination event for JOS-D1 and UCHL5, but no smearing up top. And so I think one possibility, I have no more data to stand on to prove this, but one intriguing possibility for me that I've been considering is that the E3 itself, once it transfers a monoubiquitin to a substrate, is somehow superactivated for autoubiquitination. And that would at least tie together those two pieces of data and explain them. All right, so we wanted to, in a gel, show that we could remove the ubiquitination signals from dubs. So we're, again, using our JOS-D1 and UCHL5 antibodies, and the substrates being JOS-D1 and UCHL5 in the presence of the active dubs. We saw 2-core, 21-core, and PLP2. With increasing concentration of those dubs, we removed the monoubiquitination event. In one of the dubs that did not show activity at taxon 3, we do not remove the monoubiquitination, in perfect agreement with the array. Okay, so one last piece of data before I tell you a cautionary tale, which is how I'll wind up my talk. We wanted to know, the proof of principle was with this E3 mix. We wanted to know if there was perhaps a more physiologically relevant way we could ubiquitinate a microarray. And so one thing we tried was to take a cell lysate. This lysate was provided to me by Suresh Kumar from Progenra. And you can see, first thing you see is a fairly significant background in this experiment with the FK2 antibody. That's why we're seeing some signals on the array in the absence of lysate. And I don't know why the antibody behaved that differently for me in this particular experiment. But nevertheless, the data's lost. Those signals are not going to get much darker than they are in the absence of lysate. But what you can still see through that high background is a lot of the other lights turning on, the signal, in fact, showing up. And so organizing the data for you visually, like we did before, on the left-hand column, we've got the substrates listed. Right next to that is the extent of ubiquitination in the presence of our mix, the E3 mix, and then increasing concentrations from none to 100 micrograms of embryonic kidney lysate. And going through this rather quickly, of the 35 total dubs on our array, these 6 pulled out for you plus ubiquitin were established and known to be ubiquitinated. There's literature for these 6 that already told us that these are ubiquitinated proteins, and all 6 of them showed ubiquitination plus ubiquitin. There are 6 dubs present on my array that were putatively identified by mass spectrometry-based experiments. There was never any follow-up after that, no in vitro confirmation of a direct effect, for example. Nevertheless, of those 6, 5 of them I found evidence for in this study that are, I think now, definitively ubiquitinated. The only holdout for me was BAP1, and it's not that I failed to see ubiquitination so much as it is that I had a high background for that particular protein. So I do believe that BAP1 is still a definite candidate for being ubiquitinated. I just don't have any evidence in support of that. And then finally, of the 23 remaining dubs on that array, we found significant ubiquitination events either from the E3 mix or from the HECT lysate, or usually from both. That 19 of those 23 are in fact ubiquitinated. Final piece of evidence in favor of the mono ubiquitination event for these dubs is that I also did these experiments with the lysate in increasing concentrations and detected them with tubes, and as you can see here, saw no signal. So this indicates to me that from the lysate, we see primarily mono ubiquitination of these dubs. Okay, my cautionary tale. This sent me down the wrong path for a little bit, and I hope it saves some of you some trouble in the future. Tagged ubiquitin has unexpected and unwanted consequences. This is data you saw already. If I ubiquitinate my array with wild-type ubiquitin and mock treat the array with a dub, I get robust signals. The signals are all gone from USP2 core except that dark row of spotted ubiquitin you're still seeing, but of course can't be taken off the array. That's not what I saw when I used his-tagged ubiquitin. When I used the his-tagged ubiquitin in this experiment, USP2 core, one of the most active enzymes we have, the nuclear bomb as we call it, was incapable of removing that ubiquitination event. So we repeated this experiment. We were a little bit curious as to what was going on. How is it that such an active enzyme, one that's known to act well for rhotamine, AMC, any of these assays that might structurally mimic an N-terminal tag on your ubiquitin, we know it works in those assays. Why is it not working on my array? Well, it turns out it is working on the array. When we detected with tubes, we saw, as we would expect, a loss of signal with USP2 core from the chain. In other words, USP2 core is capable of cutting that chain all the way down to the mono event, but cannot remove the final ubiquitin. And so for any of your experiments, proteomic-based especially, try to avoid a tagged ubiquitin if you can. All right, in summary, protein arrays are useful in any direction, and by direction I mean from the proteome inwards to the ubiquitome or the ubiquitome outwards. We can run them both ways. They're specifically advantageous for elucidating enzyme-substrate interaction. The facile nature of that interaction makes it almost impossible to capture by a 2 hybrid. Mass spec isn't ideal for it because you generally have to make a knockout or a catalytically inactive mutant. Not so for the array. We can use the actual enzyme. Non-core domain dubs at least appear to exhibit as much specificity in vitro as the E3s. It was a small array and a small study, but the fact that we didn't, other than the 2-core and 8-core proteins, destroy all our signals suggests that we do still maintain dub specificity in vitro. I showed you most of the dubs appear to be ubiquitinated. Specifically, I think there's prominent monoubiquitination, which is fascinating in as much as it implies that the substrate itself plays a huge role in determining the extent of its own ubiquitination. Remember, we're in vitro here. Most dubs do not deubiquitinate themselves. Next one is a little bit more, a little bit less defensive. I have less data to suggest this, but it's certainly possible that SARS infectivity is involving the activity of PL-Pro removing host dub ubiquitination. And dub activity depends on the assay. Do not use tagged ubiquitin. So the future, we need to establish the biological consequence of dub monoubiquitination. Unfortunately, I cannot tell you what that consequence is. Ataxin 3 is known to be superactivated when ubiquitinated. That one is in the literature. And I can tell you most of the dubs I've looked at do not show superactivation for activity when monoubiquitinated. So we still don't know what that is, what the consequence is. We use DubChip to establish this principle. This is the first microarray demonstration of reversal of post-translational modification. Nobody in the kinase phosphatase field has ever done this. So we've established the principle now. We think it's now time to move it to the 10,000 member protein array. And finally, all of these types of questions, I need to work with you. I'm not at the University of LifeSensors. I'm at LifeSensors Incorporated. And I can provide leads, but you need to follow them up. So let's do it together. With that, I'd like to thank you for your attention. I'd like to thank the best array company on the planet, in my experience, Gentile Biosciences in Madison, Wisconsin. Progenro, a number of my colleagues I've already mentioned, Suresh, Craig, Mike, and Jeff, and all of my colleagues at LifeSensors, Jim, Steve, Katie, Jules, and Esteban, and our newest member, Chris Wood, who didn't make the slide in time. Thank you. We have not used that now. That's a great experiment to do. Yeah. Right. Great question. Just to repeat for anybody that didn't hear it, how do you make sure that the structure of your protein is maintained on the array when we immobilize it? This is some